PCR products can be sequenced directly using cycle sequencing methods, as long as there is only one product present and the primers and excess of DNA TPS have been removed. Other components, such as comparing enzymes or proteins, could also cause problems. Therefore, the product must be purified before it is sequenced. There are several methods for purifying PCR products, like column purification, e.g., precipitation, gel purification, or ethanol precipitation. There are many methods for purifying PCR products, like DNA recovery by an electrolysis into a trap of salt, into a dialysis bag, in a membrane, agarose degradation with agarose enzyme, phenolic agarose enzyme extraction using low melting point, and magnetic bath purification with DNA fragment purification with GTG agarose and with acylic polymerase chain reaction is a powerful technique used to amplify exponentially specific target sequences of DNA. The DNA and PCR products are usually visualized by being separated in an agarose gel. DNA should be then purified to obtain specifically the target DNA fragments, or to separate the desired PCR products from non-specific amplification products and another amplification artifacts like primers and nucleotides. Purified DNA fragments and PCR amplifications from agarose gel have a variety of applications, such as cloning, labeling, and sequencing DNA. The purified DNA can also be used for subsequent PCR reactions, in vitro transcription, and restriction mapping. To purify DNA fragments which have a molecular weight less than 1 kilobase, there are many column extraction kits that could be useful because they ensure the recovery percentage up to 95% with DNA which molecular weight is between 40 BP and 10 kb. On the other hand, to purify high molecular weight DNA samples, the most useful method should be the agarose degradation with agarose enzyme, because it's the most recommended for high molecular.
For the experimental procedure, the first step was to excise the gel slice, and then we weighted it. We added gel solubilization buffer at 50 degrees Celsius, and then we loaded the column. We centrifuged it for a minute, and then we added the wash buffer containing ethanol. Then we centrifuged it again, and the last step was to add the elution buffer and to centrifuge again in order to recuperate the genetic material. In the spectrophotometer, in a reading of 260 nanometers of a wavelength, we got 0.008, and in the concentration we have 0.4015 micrograms per milliliter, and in the ratio of the readings of 260 and 280 we got 1.1519. In the first lane of figure 2 is shown the gene ruler of 100 base pairs DNA lab, and in the second lane we can observe our PCR product purified from an 0.8% agarose gel, and we can see that the sample shows some degradation, but it did migrate to the expected.
The purity of the samples were different one from another. The first sample, non-purified, had 1.875 concentration, and after purification we had a concentration of 1.15. This could mean that during the procedure of purification using the kit, some contamination like proteins or salts could have been acquired and therefore low quality sample according to the kit.
It is relevant to analyze the obtained DNA after purification to determine DNA yield and purity. For this purpose, two methods are mainly used: absorbance and agarose gel electrophoresis. In the absorbance method, you only have to use an spectrophotometer and a DNA sample in water solution. This will indicate the contamination that could be implied in the DNA with the different ratio readings that are 260, 280, and the 260 and 280. This determines the protein contamination in the sample. A good quality DNA will have an 260-280 ratio of 1.7 to 2.0.
In the agarose gel, we could know the yield by comparing the two bands, the DNA before and after the purification. This could give us an idea of the success of the purification method and the purity and integrity of the DNA base size. Our initial DNA sample had a concentration of 0.4015. This means 100%, and after purification we had a 1.008. This means the yield was of 2%. This yield is very low. This could be because an excess of agarose of gel after trimming the band containing DNA, or the use of molecular violet grade water instead of elution buffer. In fact, a heated elution buffer at 70 degrees Celsius can increase the purification yield because it will release more DNA from the gel, from the silica, and the silica membrane should be completely dry after the wash buffer with ethanol step, since the presence of ethanol can lower the purification yield. DNA gel extraction column allows extraction of desired DNA fragments only, though it is useful when needing high specificity for further analysis. Therefore, it is very important to be very focused when doing the critical steps to make a successful extraction. For example, the UV light exposure, the use of correct agarose amount, the correct selection of elution buffer, the correct washly of the column after elution, and others. All the previous mentioned steps should be followed properly to obtain the higher purification yield and the higher concentration of DNA possible.